to have you here today for the 2019 Coy W. Waller Distinguished Lecture. We're thrilled to have Dr. Bohn here from Scripps Institute as our speaker. You will hear more about her shortly from Dr. Sudesna Roy. But first, Dean David Allen will open our program by sharing the history and significance of this lecture series. After the lecture, we welcome any questions from the audience. We'll have BMS graduate students available to provide you with a microphone for your questions. It's now my pleasure to welcome Dean David Allen.
Thank you very much, Christy. Well, good morning, everyone. I'm so pleased to see everyone here today as we honor Dr. Coy W. Waller. I'd also like to thank the Department of Biomolecular Sciences for co-hosting this event, as well as Dr. Laura Bohn for serving as our 2019 Waller Lecturer. It is my great privilege each year to tell you a little bit about Dr. Coy W. Waller, an amazing man with a truly incredible legacy in natural products research. This is the 16th lecture we have held to honor Dr. Waller since 2004. Dr. Waller was born in 1914 in North Carolina. He received his bachelor's degree in pharmacy from the University of North Carolina, his master's degree in microanalytical chemistry from the University of Buffalo, and his doctorate in pharmaceutical chemistry from the University of Minnesota. In 1968, he began his 11-year tenure at the University of Mississippi as the Associate Director of the Research Institute for Pharmaceutical Sciences, and in 1970, he became the Institute's first full-time director. He advanced the Institute in immeasurable ways. His focus on natural products as a source of new pharmaceuticals laid the foundation for our National Center for Natural Products Research, the first and largest of the Institute's now 4 centers, and the only such institute that exists on a university campus. Before he retired in 1979, Dr. Waller conducted research on poison ivy and the use of cannabinoids to treat glaucoma. He also helped edit bibliographies on marijuana and cocaine. Over the course of his career, Dr. Waller authored more than 50 patents which were issued in over 150 countries. He was passionate about inspiring students and young colleagues to pursue natural products research and about helping them to realize their potential to develop new pharmaceutical products. In 1993, the university honored him by dedicating the Coy W. Waller Laboratory Complex, a natural products laboratory that houses our marijuana garden, medicinal plant garden, and greenhouses. Dr. Waller was one of the most accomplished scientists of his time. When he passed on April 24, 1998, he left a legacy of leadership and research excellence. We are incredibly grateful for his contributions to our school, our nation, and our world. We are honored to have some of Dr. Waller's family with us today. Ms. Deb Carnathan, Ms. Liz Thomas, and Ms. Ashley Couch. Will Dr. Waller's family please stand so that we may recognize you? We truly appreciate your presence here today, and we're grateful for all that you and your family have done for our school, the pharmacy, and this great university. I would like to now introduce Dr. Sudesna Roy, Assistant Professor of Medicinal Chemistry and Pharmacognosy. She will introduce Dr. Bohn and our 2019 Coy W. Waller Distinguished Lecture. Sudesna?
Thank you, David. It is my privilege and honor to introduce the 2019 Coy W. Waller Distinguished Lecturer, Dr. Laura M. Bohn. Dr. Bohn is a professor of molecular medicine and neuroscience at the Scripps Research Institute in Jupiter, Florida. She received her Bachelor of Arts in Chemistry and Bachelor of Science in Biochemistry from Virginia Tech before earning her PhD in Biochemistry and Molecular Biology from the St. Louis University School of Medicine. She completed her postdoctoral work in the Howard Hughes Medical Institute at the Duke University Medical Center in the laboratory of Dr. Mark Karen in collaboration with the 2012 Nobel Prize winner in chemistry, Dr. Robert Lefkowitz. Dr. Bohn was a tenured associate professor of the Ohio State University College of Medicine in the departments of pharmacology and psychiatry before she joined the Scripps Research Institute in 2009. Dr. Bohn has made significant contributions to opioid research and has published over 100 journal articles, book chapters, reviews, and patents over her career. Her work in bias-ligand signaling has changed the way that many researchers understand opioid pharmacology. She continues to be a thought leader in this field and is now extending the lessons of bias signaling from opioids to cannabinoids. In addition to multiple consulting roles, she is an affiliate member of the Brain Institute at the Florida Atlantic University. Among her numerous professional recognitions are the Young Investigator Award from the Society of Neuroscience, the Joseph Cochin Young Investigator Award from the College on Problems of Drug Dependence, and the Victor Mutt Lecturership from the International Regulatory Peptide Society. She also received the John J. Abel Award in Pharmacology from the American Society of Pharmacology and Experimental Therapeutics. The Department of Biomolecular Sciences is honored to have Dr. Laura Bohn with us today as she presents a talk entitled Refining Opioid Receptor Signaling to Improve Therapeutic Potential. Please join me in welcoming Dr. Laura Bohn.
I really appreciate this honor. This is fantastic. It's my first time to visit Oxford. It's a beautiful town. I'm glad there's not a football game here this weekend. My travel would have been more difficult. But I'm really happy to be here. I think I'm getting feedback. Is that okay? All right. So this means a lot to me because my research has really depended on natural products. We work now to make small molecules. But natural products are really at the heart of the receptor systems that I'm interested in. Whether it's at the kappa opioid receptor with salvinorin, or in the cannabinoid receptors with the cannabinoid compounds, or at the mu opioid receptor, which is what I'm going to talk today, where one of the longest known natural products has come from, and that's morphine. So morphine is an agonist for this receptor. So I'm going to talk about mu opioid receptors. I'm going to try to give a little bit to everybody, and I'm going to quiz all the pharmacy students later on the mechanisms. But I want to just talk about this receptor and what we know about it. So the G protein coupled receptors are 7 transmembrane spanning receptors that are found on the surface of cells, also can be found inside cells, but they're transmembrane receptors, so they span the membrane, and ligands will bind to the surface and transmit signal transduction into the cell, to the neuron to tell the body what to do. So the opioid receptor, the mu receptor, the known agonists are your endogenous enkephalins, your endorphins, but also drugs that are very useful in clinical practice, morphine, fentanyl, oxycodone. These compounds all hit the mu opioid receptor. We know from studies in knockout mouse models that if you take out the mu opioid receptor, you no longer have the antinociception of these compounds, you no longer have the overdose, you no longer have the respiratory suppression, the constipation, or the abuse liability. So this receptor is really integral to all of the effects of this receptor, to these drugs. So morphine will activate this receptor, you'll get pain relief, and I have it in green, the things we want, you get cough suppression and codeine. It's an anti-diarrheal with Imodium, but all of that in too much excess, or if it gets like Imodium, if it were to get into the brain, would have all these other problems of respiratory suppression, which is the #1 cause of overdose, as well as constipation and tolerance that we develop over time, physical dependence, to where you become reliant on the compound to not go through withdrawal, and also addiction. drugs. Morphine's a great, useful drug. If you've ever had surgery, you know this. We need this system. But we also know in recent years that this has been a real problem. And so I just put this into context. If you look over the last 20 years and the use of the heroin and opiate-related deaths, it was very low, and then the commonly prescribed opioids started to escalate. And we all know now in epidemiological studies that this tracks really to a lot of marketing, right? A lot of marketing by the opioid. I'm going to move to this slide so my microphone's on the right side when I turn my head. There we go. So as we see over time, you can see that at around 2010, we started to see a rise in heroin-related overdose deaths. And then more recently, we've seen an influx of synthetic opiates, and that would be your fentanyls, carfentanyls, that are coming in directly sold as carfentanyls and fentanyls, and also spiked into counterfeit pills of oxycodone and stuff that are sold. So this has been a huge problem. I know when I was in Ohio, we were really struck as we were looking at this, that so many people were using oxycodone. So many people were using these pain relievers. And I think that we had a dependent population that we didn't even know because regulation wasn't such that, as long as drugs are available, you don't have a problem, right? Until they started to restrict. And then that was in New York was the first to do it. And then there was Florida, where I am, and Tennessee. And as they started to implement the restrictions in pharmacy prescribing and doctors prescribing and tracking the pharmacy prescriptions across different pharmacies, so you couldn't just shop your prescriptions around and get 10 filled, well, they saw, we got a drop in prescription abuse, but at the same time, heroin came into town, and that's at 2010 we started to see the heroin overdoses. Because suddenly OxyContin, which was very cheap to buy on the street, became very hard to get, and heroin started taking place. And then heroin's quality also went very high, and the purity of the drug became very high, and we saw overdose-related heroin. And then, of course, we've seen an influx of fentanyl, carfentanil, many fentanyl derivatives coming into use. And so you may think you have your heroin that you're used to, and it has just a little bit more fentanyl, which is a very, very potent compound. And that becomes an overdose problem. So that's where we are with an overdose problem. And so the NIH HEAL initiative, if you're familiar with this funding opportunity, millions and millions of dollars have been pumped into this to try to fix this overdose epidemic. And so there are initiatives to develop new kinds of drugs and new kinds of therapeutics that won't be opioids, won't be addictive, won't have overdose, and that's great. But I'm going to talk to you today about the new opioid receptor, which is a drug we know, morphine, for a very long time. We know how to use it clinically. We know under the right conditions and situations, it's actually a very useful drug. And we're using that in our understanding of the new opioid receptor to think about how we can develop new kind of compounds that are safer and might be useful in conjunction with the known opiates. Okay, so I'm just going to break this down. So I said the target here is the mu opioid receptor, and these are expressed on the surface of neurons. And that when drug binds, it leads to the interaction of intracellular effectors. So the ligand's on the surface, it's flowing through the blood, it hits the receptor, and it turns on the neuron. It either turns it on, turns it off, it does a number of things. But this concert of assembling these intracellular signaling complexes act together to tell the neuron what to do. In pain pathways, it suppresses firing and says, don't sense this pain. Okay. That's what we want to preserve. But these receptors are expressed in many different types of neurons and throughout the brain, and they're expressed throughout the body. So the same receptor that when it's engaging in the brain that's saying alleviate pain perception is also engaging in the enteric nervous system and saying no water secretion. So that results in profound opiate-induced constipation, which becomes a problem. Same drug is hitting the same receptor in the brainstem regions that control respiratory function. And too much says stop breathing, and that leads to overdose. So we're looking for a way to refine. Now you could take an approach like Imodium was great for treating constipation because it doesn't get into the brain, you just hit the enteric nervous system. But when you have multiple effects that are associated in different regions of the brain, it's really hard to try to get the drug to just go to the receptor that you want. So we start thinking about this differently. And instead of thinking about just an agonist, so morphine's an agonist, and an agonist turns on, an antagonist turns off. And the way we used to think about G-protein coupled receptor signaling, or any of these receptors, is that an agonist would come in and we turn it on. Well, if you've ever had an apartment that you have a light ceiling fan combo, and you have one wire going to it, and you come in and you flip it on, you either have all the lights on and all the fans on, or you have it all off and you're hot. What we're really looking for is an opportunity to introduce a dimmer on each component, so that we might be able to turn up the lights and turn down the fan, or turn up the fan and turn off the lights and go to bed. In this case, we'd see maybe turn up the pain relief and turn down the respiratory suppression. And that's the goal. And that's what I'm going to talk to you today about, how we try to do this. And this concept that I've simplified is called biased agonism, or functional selectivity. And it's not just important in the opioid receptor field, but it's being applied across the board to many different G-protein coupled receptors. I just came from talking to a group in Milwaukee, and they're looking at it. Chemokine receptors, there's many opportunities, parathyroid hormone receptors, beta-adrenergic receptors, alpha-adrenergic receptors. It's where things are going. So for your pharmaceutical training and understanding, it's important to know what this biased agonism means. So it really comes to a simple concept that a ligand can bind to a receptor or a drug, and the drug binding to this protein, we know that when proteins bind to different molecules, it can change their conformation. It can restrict their movement. It can change their shape. And so the idea here is that an agonist can change the shape, or a ligand will bind and change the shape of the receptor. And we know this because the receptor will then be influenced on how it interacts with its intracellular effectors. So that is known, right? Because we know an agonist binds and facilitates the receptor to signal to the G protein, the G alpha, heterotrimeric G protein complex. Thus, they're called G protein coupled receptors. But we know also that an antagonist may be just subtly different in chemical structure than the agonist, but that shape will repel or prevent the interaction between the receptor and the G protein. And an inverse agonist will actually repel the interaction with the G protein. So when we think about these things, we know that that concept is true in the extreme of agonist and antagonist. But what I'd like to introduce to you is that there's more than G protein, and that there's different complements of the proteins that interact with these receptors throughout the body. Okay, so what if the receptor is expressed in one region, say a periaqueductal brain neuron, and it couples to certain vectors that are different than, say, coupling in the entire nervous system. Now I could hopefully take advantage of that difference, if there happens to be that difference, and engage the complex that's involved in pain pathways and avoid the complex formation that's involved in constipation. This is what we're all trying to do with biased agonism. The problem is, going forward, we don't know the individual cellular signaling pathways that receptors utilize throughout the body in their discrete little neuronal populations. And that's the challenge. So how do we even address this? So I'm going to talk today, I'm going to focus on two proteins. I've told you there's many, but I'm going to talk about two protein classes. One is the G proteins, the heterotrimeric G proteins, because it's very proximal, binds directly to the receptor. And the other are beta-arrestins. Beta-arrestins are proteins that bind directly to the receptor, therefore they represent a point right at the top that an agonist could influence binding to. The canonical model, the classic model of how receptors are regulated and signal, is that a ligand binds to the receptor and the heterotrimeric G protein dissociates, and the alpha goes off and does some stuff, the beta goes off and does some stuff, the beta gamma, the particle. I keep thinking I have a pointer. I think I do. Well, over there. That part, the G protein signaling, turns on certain processes or turns off certain processes. The receptor is then phosphorylated by a kinase called the G protein receptor kinase. And these GPCR kinases phosphorylate, and then beta-arrestin bind directly. So that's the canonical model, and it's thought that when the beta-arrestin binds, that it shuts down the receptor, turns it off, because the G protein can't fit back in there, because the beta-arrestin's there. learned that beta-arrestins play a diverse role in regulating receptors. They don't just act as desensitizing molecules, they're scaffolds. And they not only can come in and prevent G proteins, they can actually, some of them can actually allow G proteins to still be there. That work is still a little controversial, whether or not that's going to hold true for all receptors. But they can act as orienting other proteins. So the beta-arrestin may bring in another signaling molecule. And that signaling molecule may be there in one neuron and not in another neuron. Again, opportunity for selectivity. The beta-arrestins can facilitate internalization, and once the receptor is internalized, it may still signal, which allows for compartmentalized signaling, isolating a signal, or it may lead to degradation, or it may lead to recycling. So you see, if a receptor is differentially interacting with a beta-arrestin, it's a pivotal point of separating a multiple array of signals that we could either preserve or eliminate. And that's why we like to look at it. I will remind you that in addition to just G proteins, we always say mu-opioid receptor is a GI-coupled protein. Well, there's GI1, 2, 3, there's GO, there's GZ. It'll signal to those as well. And there's many different G proteins, and the expression varies throughout the body and different neurons. And the different types of G protein as well. There's the alpha subunits, and then there's the beta gamma. And it might be a little different. We look at alpha and say, well, this receptor really seems to prefer this alpha. But does it always prefer that alpha if the beta and gamma are slightly different? That work really hasn't been evaluated. But biology has done that for us, and we don't even really fully appreciate those interactions. All opportunity for refining therapeutics. This is very complicated and crowded, but I only want to bring home the point that this is a synapse, and this is showing just the number of proteins that this author wanted to talk about in this paper. A synapse is a very structurally defined area where neurotransmitters are released and where receptors are highly populated. And the proteins that are there in a synapse aren't even present in the soma or the dendrite or other parts of the cell. So this becomes another question. Will the receptor signal the same way in a soma versus a synapse? Many opportunities for protein interactions and for G protein beta-arrestin to be very differently modified response. So this is, instead of that linear simplistic model, this is my chaotic model. And if life were really this chaotic, it's a wonder we have any drugs at all that work. Because all of this isn't happening all the time everywhere. But there's the possibility, if it's ever been shown to be able to interact, it possibly could be signaling a certain way in a certain tissue. And so our job is, in pharmaceutical development, is to figure out how we can target one pathway that we want that correlates with a therapeutic benefit, and to avoid a pathway that correlates with an adverse event. And that's the challenge. And I'm going to show you studies that we've done using cell-based assay systems and mouse models and trying to make this correlation. I'm fortunate because in opioid receptor pharmacology, especially the mu-opioid receptor pharmacology, the translatability is quite good with these models I'm going to talk about as far as dose prediction, human studies, and so forth. These models are not necessarily models of pain that are great pain models, but they're good models for determining opioid receptor responsiveness, potency, and efficacy. Okay, so here's how do we start. I just told you that the body is completely complex, and there may be an opportunity to have good signaling and avoid bad signaling, but what is good signaling and what is bad signaling? And I'd say about 8 years ago, I was doing the rounds to a number of pharmaceutical companies, and they'd bring me and they'd say, which bias signaling assays do we run? And I'd say, well, what signaling pathway is mediating your therapeutic response and which is mediating your adverse event? And that's the big black box. That's the thing where we've really failed to know. And I think it's because we all started transfecting receptors into HEK cells and studying the signal that was easiest to see, rather than studying the receptor in its endogenous place and really developing our tools to analyze that more refined. But regardless, so this is how we started for the mu-opioid receptor. We started with genetic models that knocked out proteins, and in this case we knocked out beta-arrestin 2. And we used a mouse model that's been used for 100 years. You can look at the literature for analgesic responses to morphine and its derivatives. And we used those same tests very simply, and we just knocked out beta-arrestin 2. And we said, what changes? The other approach that you can take is you can take your receptor system, pick two pathways, develop ligands that activate one and not the other, and then give it to the animal in the conventional models that's known to be mediated by your receptor and see if it differs. So the opioid receptor system is great for this because most every one of the side effects that mu opioid receptor agonists have in humans are recapitulated in the mouse. It's great, diverse physiologies. Okay, so the first study, this was 20 years ago, beta-arrestin 2 knockout mice. We gave them morphine, and they had an enhanced response. So this is a Hotplate Test. I'm going to use it throughout the talk. Basically, the higher the number, it means you give them the drug, you put them on a warm plate about 55 to 52 degrees, so hot sidewalk. And you put your foot on it, you go, oh, you know, running across the pool platform in the summer, it's hot. It's not so hot it's going to damage, so we put a cap on it and say no longer than 20 seconds. We say, how long does it take? So down here at 0, it took 5 seconds, and up at the 100%, it took 20 seconds. And so we measure each dose in each animal for that. And so we found that morphine worked better if we took out beta-arrestin 2. And that really matches that first hypothesis of how beta-arrestins work. Think of the word beta-arrestin. It's a brake. It's a resting signaling. And so you took out the brake, and the drug worked longer and better. We then chronically treated those animals, and we gave them morphine daily, and then if you do that, you'll get a shift in potency, and that's tolerance. So what used to give you full analgesia in day 1 in the open circles, after 9 days of treatment, it requires a lot more, about 4-fold dose to get the same kind of pain relief. And that nicely models the human condition as well. We increase dosing over time so you hit the pain relief threshold that you want. The animals that lacked beta-arrestin 2, there was no shift. It was a very small shift. So they didn't develop this tolerance, which again suggests it's like a brake. Take the brake out of the system, the receptor keeps signaling. Well, we worried about that because we said, will they have overdose more readily if we took out the brake? And we were surprised when we did these studies in mice and looked at their respiratory. This is their breathing frequency. Okay, so this is a plethysmography study where the animals were measuring breathing. You can see where morphine is injected. And the one that dips down, the white open circles, that's the wild type. That's a typical response. They decreased breathing. But the squares, they didn't change. They stayed steady. Or the filled circles stayed steady. And that's the knockout. So they were actually protected against this respiratory suppression. And that was enough for me at the time looking at this, like, okay, there may be a benefit to developing a compound that would signal through the G protein pathway, keep that all going, and not recruit this beta-arrestin component, right? Because maybe we could have something that was efficacious for treating pain, maybe not develop tolerance, maybe not have respiratory suppression. At the time we were doing this, this was in collaboration with Bob Lefkowitz at Duke. I went off and started my faculty position, and he started the company Trevena. Trevena has a compound that's in clinical trials, but you know, this is the benefit of staying at a problem in academics because you can learn more about your problem before you launch into therapeutic development. And I know that their compound is not quite meeting thresholds where it needs to be, but they did see a slight benefit in eliminating some of the respiratory spread. They didn't eliminate it, they just got a slightly better, slightly not significant better therapeutic window. So what they did and what we have done is develop compounds that stimulate G protein signaling and avoid this beta-arrestin recruitment. And we published this work in 2017, and I'm going to go through it kind of quickly, the development of the compound and some of our data. And for the chemists, there's our structure and all the things that chemists and anybody who's doing therapeutic development, compound development, plays well with Lipinski, has straightforward synthesis, easy scalability, very nice, easy to work with compounds. We're able to generate quite a bit in the lab. Everything I'm going to go through quickly, but we've run a lot of controls. We use mu opioid receptor knockout mice for control run through the psychoactive drug screening program for off-target, and NIDA has now run it through their medical development branch to make sure at Eurofins panel that it has very good selectivity for the mu. That's important because we're going to take compounds in vivo and we're going to try to define some property of it that gives a difference in physiology based on bias signaling at one receptor. But if the compound hits another receptor that causes a different effect, that would confound our understanding. So that's why it's been very important. I don't show it all here, but we really make sure the compounds are selective. I'm going to talk about biased agonism. I'm just going to briefly introduce this topic. So when I look at this, and we're going to study how a compound will activate a G protein pathway versus an arrestin pathway. And it's going to be a bunch of cell-based assays, and you're going to see a lot of nonlinear regressions. It's very important in your drug development, your SAR development, your structure-activity relationship for chemistry, that you know a parameter that you're going to uniformly trust between all of your assays. Because this is a campaign we've been doing for 9 years. And so these different people come in and different people run the assay. And so the assays have to be stringent and they have to be controlled. And in order to compare the effect in one assay versus another assay, we utilize what's called the operational model, which was proposed by Black and Left, receptor occupancy versus activity, to understand how a receptor signals in one way relative to a reference, so a known compound, and then we compare it to how that same compound performs in another assay, also relative to a known compound. And in that way, we were able to normalize its individual effect between the two pathways, and then make a comparison. Otherwise, you can't make the comparison. And I use the example of an enkephalin or morphine, and I say, what's the potency of morphine? And what is the potency of morphine? And your question should be, in what assay? In what condition? Is it a patient, or is it a dog? Is it a cat? Is it a mouse? Is it a monkey? What are we talking about here? And it's the same thing. If you were to test morphine in a GP-gamma S binding assay in a CHO cell that's transfected, you're going to get a potency around 30 nanomolar. And if you were to test that same drug in the same assay but using a brainstem membrane, you're going to get about a 200 nanomolar potency. So if I were to say, oh, those are two different assays, and look, it's 10 nanomolar potency here, and it's 200 nanomolar potency here, it's biased. No, it's the system. The system is changing, and when the system changes, the drugs may look different. So what we do is we take a drug, in this case the reference agonist, we use an enkephalin called DAMGO, and we use it to define the system. And so DAMGO here gives a full response in our assay. This is a GTP-gamma S35 binding assay, and it gives the full response, and here's morphine compared to it. Well, morphine we know is a partial, but in this overexpression system, it's marginally partial, it's 80% efficacy, and its potency is about twofold shifted. Over here we have a beta-arrestin recruitment assay. This is an enzyme fragment complementation. That means part of a galactosidase is on the receptor, part's on the beta-arrestin. They come together. 90 minutes later, you add a reagent. The students love it because it's quick and it's 384 well, and you get tons of data. But that's how morphine looks. And in the early days when people started doing these experiments, they said, oh, morphine's a biased agonist. Oh, well, morphine is different than DAMGO, but it's a partial. The potency shift is similar. This assay picks up partiality a lot more than that assay. If you did this in brain, morphine would look just like this. It would be very partial, where there's not as many extra receptors. We did this comparison, and we applied this modeling to say here, morphine was balanced. It was not showing bias. Now, I could have run this assay 10 different ways, and I could pick the different reference compound, and I can tell you, yeah, morphine's biased. Because bias number, just like potency, will change. Because bias, what is bias? Bias is a comparative term, right? It's not an all or none. So it's compared. It's more or less of this or more or less of this, right? And so if you change what you reference or you change the system, yeah, bias is floating. So there's no absolute. So that was one of the first challenges we did with this. And we implemented this stringency because we already believe morphine's a good molecule, and we want to be better than morphine. So, I'll just show you briefly buprenorphine. This is another one. Buprenorphine's a partial in both. But over here in the arrestin assay, it looks flat. And you'll see a lot of this. People will propose, oh, it has no response. We can't even calculate bias. So therefore, it's ultimately biased. It's extreme bias. But we know as a partial agonist, and for students, for partial agonist will inhibit a full agonist down to the point of its own agonist efficacy, right? So you can use that property. It's demonstrated here in the G protein on top, that if you put the buprenorphine against DAMGO, it brings its signal down like an antagonist, down to the same level that it would signal on its own. In the arrestin assay, now we can actually capture a functional affinity of buprenorphine for a beta-arrestin bound receptor, and we see it actually has quite good affinity. And we feel that perhaps a biased agonist shouldn't have functional affinity for the receptor coupled to the beta-arrestin. And so in our hands, we would say this is not the kind of biased ligand we're looking for. And when we do our calculations, it indeed doesn't differ from zero, so we would say it's not biased in these two assays. Okay, so that's my big spiel on biased agonism. I think it's important to understand that because it's all across GPCR receptor pharmacology and therapeutic development. It's very popular. So bias factor is not a magic number. It's a number that you calculated based on what you used as your assays and what you used as your reference in all of your conditions. And in most cases, people don't even take into consideration kinetics, which I think is the new way that we're really thinking about this, because kinetics can mask all of this and make something look biased when it's not. So keep it in mind, and changing the context or changing the reference will change the perception of bias. I would also point out that there's flavors of bias. And what I showed you with buprenorphine, it doesn't mean that buprenorphine is not going to be a good drug. It may do things that we really want it to do, but maybe it's doing it because of its partiality, the fact that it's such a good, potent partial agonist, and that under certain tones it can drive things down to normalcy. So this is an idea that there's not just going to be, this is a biased compound, ergo it should not have this side effect or it should do this. There's going to be flavors within this depending on how we work. This was important for us because we're working in structure-activity relationships, right? We're starting with a few compounds and we're going to build many, many compounds in the group, and we're going to do comparative pharmacology. These are the compounds I'm going to talk about. So we have this 800-some compound collection that we've characterized, and at the end of, at some point you have to stop and say, what do we want to go after physiologically? We have all these compounds. So what we did is we selected 6 compounds from the collection that span bias. Some that preferred beta-arrestin, and increasingly preferring G protein over beta-arrestin. And then we subjected them to full analysis in vivo through correlation. So this is what G protein looks like on the top. They're just graphed on different bar graphs or on different graphs so you can see them. They're all good. They're as good as morphine, so all the potencies are good as morphine or better. A couple of them were full, most of them were partial. And then here's the arrestin. You can see we like beta-arrestin and we go to not doing beta-arrestin. And I'll point out that our curves aren't flat and that they're not hiding partials, because if we put it against DAMGO, it doesn't displace. So they really have very low functional affinity for beta-arrestin bound opioid receptor. We calculate then EC50s, we calculate the maxes, and then we calculate the bias, the delta tau over Ka. And really the thing to take home here is that we have this gradient of compounds, and morphine's right there in the middle. And the ones I'm going to talk about the most are the ones with the arrows. And I chose them because they were the closest to morphine. Well, morphine has an arrow. Okay. We did a lot of controls. I said I'm not going to go through them. We checked everything in the discovery phase was all against the human opioid receptor. The mouse opioid receptors was only 2 amino acids shorter at the N-terminus. So we ran everything in mice too, and mouse receptors, and mouse cell-based assays, and orthogonal screens. We did all of it, read paper. And we did all the knockout mice, and we did extensive pharmacokinetics and microsomal stability. And the reason we do the PK is because I'm going to take a compound now that we've characterized as cell-based assays, and I'm going to give it to a mouse, and I'm going to say, do you work the same as morphine? But if my compound's metabolized in 10 minutes, of course it won't produce the effects that morphine did, right? So we had to make sure it also got into the brain. It has a very nice plasma distribution. It lasts for quite a while. has very high albumin protein binding capacity, but it gets into the brain at 6 to 12 micromolar. So it gets into the brain a lot. And if we do the calculation for free fraction, it's very equivalent of the doses we're using to morphine. Okay, so I'm just going to show you some behavior data. This is that same hot plate that I showed you before, and tail flick on the bottom. It's basically a tail and some warm water, and you just time how long it takes till they lift it out. The higher the number, the more antinociceptive the compound was. So this is all showing different doses, but the maximum efficacy dose for each of these compounds. They all produced antinociception, and then we took them into a mouse pulse oximetry. So this is just pulse oximetry to measure O2 saturation of arterial blood. And opiates dramatically will drop O2 saturation and suppress breathing frequency, which is shown on the bottom. I think it's clearer to see in the O2 data. The fentanyl in the compound 11501 at the same doses that produce maximum analgesia are producing massive respiratory suppression. Those are the beta-arrestin preferring compounds. As we go this way, we get to our G protein preferring compounds, and we start to see a loss of the respiratory suppression. Now, you can't believe anything on a single dose, so we did full dose response analysis, which took a tremendous amount of time, so I show this picture because it represents a lot of work. And every one of them has a knockout mouse control in it at the highest concentration. But what you can see from a mosaic, even from the back of the room, is that the top two graphs, they all look similar. We get a dose response for antinociception. But if you go across the bottom two rows of graphs, everything is changing. We have a lot of respiratory suppression and a lot less as we go this way. And the way I've laid it out here is increasing the bias factor, of increasing the difference between the G protein preference and the beta-arrestin preference. We then can calculate a therapeutic window by calculating the ED50 of both responses, and that's shown here, and I'll just quickly show you, here's how the ED50s look, and this is the therapeutic window. So we take the ED50 of the O2 over the hot plate response. Now we start to get a spectrum. Okay, very simple. So we noticed that this was, of course, correlating with the bias factor. So we did a correlation analysis of G protein versus arrestin and respiratory over antinociception, and we had a very nice correlation. And that's really the best we can do at this point. To know, is our SAR, are our cell-based assays helping us pick the right compounds that will widen our separation between antinociception and respiratory suppression, which I think is a pretty good goal. We also found in our data that if we took the beta-arrestin preferring compounds and looked at those individually, we had a nearly perfect correlation with having a very narrow therapeutic window. So I think that if we had done sufentanil in the mice, it would have fallen right in here as well. And I think most of the carfentanils would. So I think that's an important thing to consider too if you're doing opiate drug development. You might want to use these assays in a predictive manner. I will also point out that just because it has a bias factor of 30, these two have a similar bias factor, it doesn't mean the compound's going to be devoid of the side effect. Okay, I've shown you the range, and what sometimes gets lost in that comparison is that even though the compounds that showed a lot of bias, they still produce respiratory suppression. It's still here. It's just the window was a little wider. We could get efficacy in pain perception before we hit efficacy in respiratory suppression. So this compound over here, the 14968, is very potent. It has 0.44 mg per kg ED50 in the hot plate. And the other, 15098, is very close to morphine. It's around 8 mg per kg. So you can see an effect at the doses, but the window was better for both of them. They were actually very similar. Okay. So potency and efficacy will still matter in each pathway. And if you do get a biased agonist, don't expect it to be completely devoid of side effects, right? Because if we think about those protein-protein interactions, it's just overcoming thermodynamics to have an interaction with the secondary effector. It's protein-protein interactions. Ligand binds, it has a nice thermodynamic, can interact. Ligand bind, less energy put into the system, so maybe it doesn't interact, but enough of it and enough other signals are going on, you may not even need that other protein to facilitate signaling. Getting a little waxing poetic. So I'm going to show you some data that just got published. It's just been accepted. And this is looking at tolerance. So I showed you if we chronically gave the compounds, we could avoid tolerance in the beta-arrestin 2 knockout mice, the morphine. And so I just want to talk about some new data. So this is the same kind of assay that I showed you before. This is the beta-arrestin 2 knockout mice with morphine. They didn't develop very much tolerance, just about a 1.3-fold shift, very little, in the Hotplate Test. We ran into a problem, and anybody here doing delivery and will recognize this problem, that our compound was not soluble for repeated IP dosing. So we realized we always measure PK, we always draw blood to make sure our chronic dosing, we were keeping our drug steady state. And 3 days they looked great, and then the 4th day there was no antinociception. Whoa, did they develop tolerance? Well, usually tolerance is gradual, and this was nothing. So we did gross spectroscopy and looked, and we saw the compound was falling out of solution in white pellets. So anybody who does formulation will appreciate this. We tried many different ways to overcome this, but you make a compound that turns out to have 70% oral bioavailability, you don't mess with the chemistry. Because this compound actually did turn out to be very orally bioavailable. And this is showing a PO dose versus an IP dose. It's beautiful, spot on. And so we get a similar ED50. So we decided to go to an oral dosing procedure for the 1718 compound here. We did the PK. This is a BID dosing paradigm where we treated them every day, every 12 hours with half the dose. So if they were getting 48 mg per kg per day, they were getting 24 in the morning, 24 in the evening. We did that for 7 days, and we measured blood draws at 2, 6, and 12 hours after doses on 3 days to show that we were maintaining a high level of compound delivery. Morphine is shown here in an osmotic mini pump because morphine is not very bioavailable orally in a mouse. It's only about 14%. And so it's best to give morphine by an osmotic mini pump, and that's showing the levels here. Oxycodone, though, is orally bioavailable, so we used it as a side control and gave it in the same exact way that we gave the 1718 at a total of 24 mg per kg. And you can see, in comparison, you get a lot less oxycodone available and its metabolites than you do 1718. So keep that in mind. We're going to hammer our system with these animals and show very high concentrations over several days, much higher than what you'd see with oxycodone or morphine, and ask, do they develop tolerance? So this is morphine pump, and this is well known in a mouse. At 24 and 48 mg per kg, we get 2- and 4-fold shift in tolerance in the potency. For oxycodone, we just did 24 mg per kg orally BID for 6 days, measured on the 7th day, and we see about a 3.3-fold shift in potency. So yeah, we get tolerance. This is not news. This is known. This is 1718, and I should fix that top point because the closed diamond is up there. They do reach max. Very little shift in their antinociceptive potency. And in fact, it was, again, a 1.3-fold shift, which I thought was funny because that's what we saw in the beta-arrestin 2 knockout mice. Oops, I have oxycodone there. And that's showing the shift in potency at each of the doses. So we were really resistant to developing tolerance in this assay, which I think is cool. We then did a study where we took out the pump and we stopped dosing and we started monitoring withdrawal. So when you chronically treat a mouse with an opiate and you take away the drug, they go through signs of withdrawal. And those can be counted and scored in detail in the paper, which is now available. It's essentially very obvious. Everything I should mention is extremely blinded. It takes about 5 people to do all the different kind of blinding and dosing and scoring so nobody knows what they're looking at. And we always run a saline or vehicle control alongside all of these studies. So what we found is, yeah, after chronic opiate treatment, after whether it's SR or morphine, the mice go through withdrawal. There is withdrawal. We are encouraged that it didn't quite seem as severe, and it seemed to go away faster. It was done within 2 days, whereas morphine persists for 3 days. We don't know if this is a properly biased agonism. We actually think that this is a benefit of our slow PK. We have a nice slow off and clearance and high binding. So maybe it's nicer to taper off of a drug, like an opiate, which we know in the field, that a slow looked at cross-tolerance, and if we chronically treat with morphine and then try to test SR, well, they're tolerant to morphine, and so they're also tolerant to the SR treatment. If we treat with the SR compound and then treat with morphine, well, they weren't tolerant to SR, they're also not tolerant to morphine, so they're still responsive, and that's quantified. So then we had these mice that we chronically treated with morphine, and we gave SR, and so what happens the next day? So that's the 50% point over here, that day, that 1 hour, that's the first test. And so, okay, they're 50% responsive. And then we said, next day, we gave them the drug again, and they seemed to be getting a little more responsive. And by 48 hours, we're giving it again, and they had restored responsiveness to the SR compound. Well, that's weird. We're showing here buprenorphine in the same type of setup. These are morphine-tolerant mice. We start giving a very low dose of buprenorphine, not really an analgesic dose. We gave it for a couple days. Nothing got better. So then we said, what about after 3 full days of doing this, and then we challenged them again with morphine? And what was really surprising here, well, it wasn't surprising for buprenorphine in the orange, is that the animals that had morphine and were morphine tolerant, and we substituted buprenorphine, they were still tolerant to morphine. I don't think that's a surprise. But when we gave the SR compound for 3 days, they restored their potency to morphine. And that, I think, is the surprise. I think that's very unusual, that an agonist could come in and interact with the receptor and actually restore its responsiveness to morphine after it had been desensitized. So it's very exciting for us. And we also found in our control study where we had vehicle-treated saline pumps, and then we gave them buprenorphine for 3 days, that was enough to induce robust tolerance. So buprenorphine is currently one of the top therapies for opioid dependence and substitution therapy. The problem is the system stays desensitized, and I think that might be an opportunity that we can pursue with compounds of this nature to restore potency and responsiveness to morphine. What was nice about those animals is when we did this study again, and this time during those 3 days that we were treating the morphine-dependent animals, intolerant animals, to suppress or to reinstate the morphine sensitivity, we also monitored withdrawal signs. And you could see that while the drug was on board, it was not only resetting the pain perception, but it was suppressing the withdrawal, which is what you'd expect a normal opiate to do. Methadone, buprenorphine, rotational therapy will suppress withdrawal. So we think that there's potential in drug development for compounds of this class. We're actively looking at the molecular pharmacology to try to understand what is it doing to the receptor. The paper has a little bit more biochemistry in it. We're actively looking at this in different brain regions and neuronal populations to see what is it doing to stabilize that G protein signaling state, and is that what's really leading to this revived receptor system. So the hypotheses and conclusions going forward are that receptors can signal differently and be differently regulated in a context-dependent manner, and that we can hopefully take an opportunity to develop therapeutics that will broaden our ability to treat disease without side effects. Bias is not independent of potency. The best we can hope for, I think, is a wide therapeutic window, not an all or none. It's not going to be magically we get rid of all the side effects, but hopefully we can refine the therapeutic. And I think that maybe we can lead to a differential regulation and even reset receptors, and I think that holds tremendous potential. So I've showed you all this, and I showed you we started our whole drug screen with G protein versus beta-arrestin, and I showed you beta-arrestin to knockout mice didn't have some of these side effects, but I haven't shown you that beta-arrestin does any of those side effects in vivo. And that's where you guys, the next generation, come and work on these things, because it's really hard to show this signaling in the endogenous setting of the regions that regulate each of these side effects and pain responses. But this is our current hypothesis, but it could just as well be another protein that we're not even thinking about. Because even though beta-arrestins gain all the fame, there are a lot of regulatory scaffolding proteins available for the receptor to interact with that we may or may not be engaging in the system. Okay, so that's reality in any one of those. So I'll just end with showing my lab. This was taken recently. We We have a very good group of people. They're all wearing their lab coats because the Institute was taking this picture to show how good we are. And we all have our safety glasses on. So, yeah, we were doing everything we should be. And yeah, so the work I talked about today was spearheaded by Colin Schmidt. She really organized everything. She had been a graduate student with me at Ohio State. She's not in the picture because she's gone off to Biogen where she has a really nice job. But she stayed with me as a postdoc. She did her PhD in serotonin receptor signaling. And then we switched her so she'd have more of an industry experience because she wanted to go into industry. Travis Grimm, who's not in the picture because he just left, he went to a pharma in Cambridge, but he did spearheaded the tolerance studies with Fanny Pantuli, who's here. And we continue to do studies in the opioid field, the mu field, and looking at reward pathways, of course, abuse liability, and so forth. And we have an entire program on kappa receptors, which I did not show, but we have very nice compounds there and correlating physiology separating. And of course, we have a cannabinoid program, and I will just pitch that. I have postdoc openings, I have graduate student openings. We're trying to work, and there we take more of a structure function, crystal structure, cryo-wave structure, predictive chemistry to try to model in. I think our first shot on goal is selectivity, and then hopefully some biased agonism. So, thank you for your time. And I'll take questions.
Yes, thank you, Dr. Bohn. That was fabulous. We'll open the floor for questions now. The students have microphones. If you just want to raise your hand.
You can ask me if it's addictive. And I hope you're at least wondering. I think it's on. I can hear you.
Professor Bohn, that was fabulous. The story is complicated enough with respect to bias signaling. When you throw in receptor selectivity and the capacity of drug molecules to interact with other GPCRs, it's got to be confounding. And particularly with morphine, where its metabolites themselves, one in particular, has its own sets of analgesic properties. Can you comment on that? Thank you.
Yeah, it's funny because I just got NIDA to send me morphine 6-glucuronide, morphine 3-glucuronide, and that's important because the mouse doesn't even make 6-glucuronide, which is the very potent molecule. So we've actually just started putting that into our screens. Very interested in that, yeah. So we don't know what it's going to do yet, but I expect in most assays it turns out to be very, very potent. And I expect it to actually probably perform a little better even in the arrestin assay. Yeah, and that's another compound of like oxycodone. I didn't show any screening data on oxycodone because oxycodone doesn't do much at all to transfect the cells. It's the metabolites, right? So, but yeah, it's very important. I think also the cannabinoids, that's where we're really running into the big question. It's, just give me something selective. I can't take it in vivo until I know. I can't test bias agonism in vivo yet.
Questions? First one, thank you.
Yeah. I don't think it's on.
Hold it closer to you.
Hello.
Yeah.
Yeah. Thank you for a fabulous presentation. I was just interested in the concept of ion channeling receptors with respect to bias signaling. Is there any future avenues we can dig into this ion channeling receptors, especially with the barbiturates, GABA signaling pathway, where we have a lot of respiratory depression coma and then at the highest doses? So, is there any way that we can just probably?
Yeah, absolutely. And I think this is applying. I mean, they're looking at it at receptor tyrosine kinases, hormone receptors, nuclear hormone receptors, ion channel receptors. There's emerging evidence of this kind of bias bias, selectivity, right? I think in the ion channels, though, we might see a little bit more of allosterism, and maybe the allosteric modulation shuttling it more for one pathway or another, intracellular binding lipids, phospholipids in particular. I think that all that's really connected, right? I know that the group in Monash, they like to say bias agonism is just allosterism, which in a sense it is. It is. It's being preferentially engaging in one pathway or another is due to some sort of cooperativity, right? That's where the context comes into play.
Okay, if not, as a token of our appreciation, the University of Mississippi would like to present you with the Coy W. Waller Distinguished Lectureship Award, and this is in recognition to your leadership